So, in this last lecture on robustness and evolvability, we will closely study the concept of neutral networks, which are very important for our understanding of robustness and evolvability and how they can coexist in biological systems, and also discuss genotype phenotype mapping, which is central to how we build these genotype networks or phenotype networks and the corresponding neutral networks. Let us try to understand the concept of neutral networks. This is very central to our whole understanding of robustness and evolvability in biological systems. So, you now start imagining a genotype space, right, where you have where what I show here on the left is a genotype space. So, every circle, every yellow colored node here is a genotype, and two nodes are connected by an edge if they can be reached via a simple mutation. So, they are genotype neighbors. So, if you want to have a quick example, let us consider an RNA sequence of this sort, right. It is a very small RNA sequence. If you did point mutations, you can get many more RNA sequences, right. So, how many RNA sequences can you get from this? You can have 3 variations here, 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 you can have 3 variations here. So, what are the total number of possibilities? 3 plus 3 plus 3 plus 3, right. You have only 15, you can't combine the two because that would be more than one mutation, right. So, before we get there, what is the total number of, let us say you have an RNA sequence of length L, what is the total possible number of such RNA sequences? How many different RNA sequences of length L can you have? 4 to the L, right, because you can have A, U, C or G in position 1 into A, U, C or G in position 2, so on till position L. So, that is 4 into 4 into 4, 4 to the L. So, you can imagine the space is massive. If you are talking about 30, an RNA sequence of length 30, you are talking about 4 to the 30 or like 2 to the 60, which is basically going to be somewhere around 10 to the 18, very massive number, right. But whereas the number of neighbors are only 3L, because you have, so you have A here, you can have C, G or U here, you have U here, you can have A, C or G here and so on. So, each of these would be a separate sequence, you have a total of 3L sequences, 3L neighbors, in this case it is going to be 15 neighbors. So, if this were the genotype space for size 5, how many nodes would we have? 4 to the 5 or 2 to the 10, which is 1024, you will have about 1000 nodes here, right, and how many edges will you have? Every node will have 15 edges to each of those neighbors, right. So, you will have a regular graph wherein there are 1024 nodes, each with 15 outgoing edges. So, the total number of edges will actually be 1024 into 15 by 2, because you will have them both ways, right. That is going to be a reasonably dense graph, might look somewhat like this. This is nothing but the genotype space. Does it make sense? You understand what a genotype is, like DNA or RNA or any underlying aspect. You could even think of it as a network topology, we will come to that in a moment. But genotype is the underlying genetic makeup of an organism, so whatever that is subject to change. Phenotype is a sort of higher level observation, right. So, it is a trait, for example, right, can an organism grow in a particular environment or not, what is, or hair color, eye color, these are common phenotypic traits, or it could be say RNA structure, protein structure, so on and so forth. So, all these could be potential phenotypes. So, now let us take this genotype network and map the phenotypes onto this, which means we essentially color up the network. So, every color now represents a particular phenotype, right. So, if you see a red color, all the red nodes together form are like the same phenotype, all the violet nodes are one phenotype, all the grey nodes are one phenotype and so on and so forth. Now, what is a neutral network? These are networks of genotypes that share the same phenotype, meaning if I pulled out one color out of this graph, that would be a neutral network. For example, this is the largest neutral network on this graph. It is technically a set, not a network, because you have certain nodes that are disconnected and so on, but you can see that this is a network. What does this network mean for us? What is a node again? It is a genotype. Do all, and what is unique about all these nodes? They share the same phenotype, right, which means that if you are here, if you start on this node, you can mutate here, then mutate here, then mutate here, here to any of the neighbors without changing your phenotype, which what does that mean? It means it is robust. It can change genotype, but the phenotype really does not change. So, that phenotype is actually robust. This is a smaller neutral network, incrementally smaller fragmented neutral network, and this is just multiple neutral sets, disconnected multiple neutral sets. So, what implication does this have? Right. So, you can see that neutral networks just completely lace the entire genotype space. So, there are the genotype space is basically overlaid with several neutral networks, and these neutral networks have implications for both robustness and evolvability. We will see how that is. So, let us first understand the genotype phenotype mapping, because that is central, because if you have to think about what a color means and so on, you need to have a mental picture of what is a genotype and what is a phenotype. So, a very straightforward example of genotype and phenotype is sequence and function. Function is usually harder, so maybe sequence and structure as you see here. So, RNA or proteins, you can think of sequence being the genotype and structure being the phenotype. For a regulatory network, you can think of the regulatory interactions or the regulatory topology being the genotype and a gene expression pattern being the function. For a circadian oscillator, I am giving these examples because these are from interesting published studies in the past. So, for circadian oscillators, the regulatory interactions can again be the function and be the genotype, and whether or not an oscillation is observed can be the phenotype. And we have also studied digital circuits in the past, wherein the circuit or components can be the circuit components or the wiring can be the genotype, and what is the Boolean function that is computed by that circuit, that can become the phenotype, and then we can try to design a robust circuit and so on. Similarly, here you can try to design a robust oscillator and things like that, but it is little harder if you are looking at say protein protein interactions or signal transduction and so on. It is difficult to really map what is a sensible genotype, or it is the genotype is maybe not that difficult, but how do you map the phenotype? And to add to this, one can really imagine metabolic networks where the presence or absence of reactions can or genes can be construed as the genotype, and phenotype can be computed using flux balance analysis, right. You can just do an FBA simulation to see if the organism grows or not and use it as a phenotype. So, this mapping may not be straightforward always, it could be a little tricky. So, let us look at how neutral networks play a role for robustness. Do you see any similarity between the two graphs? The nodes are actually the same, the number of nodes are actually the same, whereas the network on the top has far fewer edges than the network on the bottom. You can clearly see that the average degree of the network on top is much lower than the average degree of the network in the bottom. So, now let us imagine all these colored dots that you see there, let us just imagine that they are organisms evolving on these neutral networks. So, what does evolution mean here? What happens when a mutation happens? It will move along an edge. Can it move outside the neutral network? Yeah, because this does not account for the entire space, this is just a neutral network. If this was a genotypic space, then you are it catalogs all possible mutations that can occur. This is only a subset of the genotype space, essentially a network, a layer of the same color that we saw in a couple of slides ago, wherein you start with these with some node here, and you can take any of the edges. There are some potential edges that are not on this as well, but what is the issue with those edges? Taking those edges will change your phenotype. Perfect, very good, right. So, now if you let them evolve for a while, what happens? You see that here there are many more edges that are taken which take you out of the neutral network, right, and for all you know that could be a lethal phenotype, right. So, let us assume it is a lethal phenotype and those organisms die, which is indicated by the small red arrows that you see, and if you keep proceeding you will see that all these in the neutral network on top the organisms are still stuck around wherever they started more or less, whereas here they have been able to percolate to distant parts of the genotype space. Like in this neutral network you are very likely to have more deleterious mutations, whereas if you have high robustness you are more likely to be able to explore the space, and as a consequence you might encounter novel phenotypes. We will see what that is in the next slide. Here, so you are talking about the last point that I made. So, robustness and evolvability both somewhat correlate with neutral network size and connectivity. Size alone is not sufficient, connectivity alone is not sufficient, as you will see here. Okay. So, this is a network which is highly connected and but somewhat very tightly connected. This is a network that is completely disconnected. This network is intermediate. It is it is well connected, has what is the important parameter, network parameter that differs between these three? Average degree maybe. What else? Another very simple parameter. Clustering, yes. More obvious, clustering does vary, I agree. Most obvious, diameter. Diameter here is essentially 1 or something. Here it is very large, could be like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 or something like that at least, right. Whereas here the diameter is very small, it could just be 1, 2, 3, 4 or something like that, right? And of course there are disconnected sets and so on. So, what does this mean? So, here if I start off on this genotype, I can never reach the red phenotype. Here if I start on this genotype, I can again not reach the red phenotype at all. Whereas here if I start on this genotype, I can have several neutral mutations that can finally take me towards red, right. I can reach the red phenotype here. So, this is what one calls a novel phenotype, potentially a novel phenotype. You could argue that this could be a lethal phenotype as well, but the thing is you have access to several more phenotypes in the neighborhood. If I look at, so if I were to look at the entire space like in the previous figure or even here, right. So, if I am on these nodes on this large neutral network, I can probably move a lot and suddenly I find myself close to so many other phenotypes, or if I go to this region of space which is still accessible through neutral mutations, I can again access many more phenotypes, or if I go to this region of space I can still access some more genotypes with different phenotypes in the neighborhood. Does that make sense? So, all this has implications for robustness and evolvability. I would say robustness is nothing but average degree. That is not necessarily robustness, right, because robustness is the ability to maintain the same phenotype. Evolvability would be that. Evolvability is the ability to access new phenotypes in the neighborhood. So, robust phenotypes tend to have higher evolvability, meaning if you are robust like this phenotype, you could have access to more genotypes with different phenotypes in your neighborhood. What do you mean by quickly? If I had other nearby genotypes with different colors, that is still going to be reasonably good, that is still going to be evolvable. Here this is not evolvable because this is just, this does not seem to be connected to any other phenotype at all.
If it had phenotypes, like if it had like multiple red phenotypes all around it, then would it still be robust?
Yes, assuming that this is still the network, you still have enough phenotypes, enough genotypes that share the same phenotype, right. The size of this neutral network is still large. So, you can still accommodate several mutations, whereas here you seem to be able to accommodate even more mutations, right. If you count the number of edges here, it is clearly higher.
So, if it is more likely to stay within the same phenotype, it is robust, but it should also be able to go to another phenotype.
Yeah. But you can imagine that if you have a network that is very large, then very likely in most genotype spaces you are going to have access to new phenotypes in the neighborhood, because as we saw, the way at least we defined the genotype spaces and so on, they are pretty regular graphs, right. So, every node is going to have n number of neighbors, and if some k of them are the same phenotype, that is your average degree, that is going to be your robustness, but the rest of them are going to have different phenotypes, and for a larger network the neighborhood size will be even larger. So we look at those definitions in a moment. And populations evolving on large neutral networks have greater access to variation. This is a large neutral network, so I have good access to variation. I can just move all around this place and suddenly come close to a red phenotype or some other phenotype and so on. So the fact that this neutral network is structured thus becomes very important. Right? What is the structure? So you just can't look at if you look at the number of nodes, it's the same in all cases. Right? If you look at the number of edges, it might be somewhat similar here, but you also need to look at the diameter and the connectivity and so on and so forth. So now let's look at some definitions, some interesting definitions. What is genotype robustness? Right? So it's the number of neutral neighbors of a genotype G. Does it make sense? So number of neighbors that share the same phenotype, meaning these are the edges. If I'm at a particular genotype, these are the edges that I can take without disturbing my genotype, without disturbing my phenotype. So what is phenotype robustness? Is the number of neutral neighbors averaged over all genotypes with a given phenotype. So this is nothing but the average genotype, average degree of the entire neutral network. Does it make sense? What is a genotype again? It's a node. Very good. What is a phenotype? It's potentially the entire neutral network. So if you want now compute robustness of the phenotype, you essentially look at the entire neutral network, right? So what is the average degree across that neutral network? What is genotype evaluability? It's obviously the number of different phenotypes found in the immediate neighborhood of a genotype, or essentially the number of non-neutral neighbors. You are right. So genotype robustness and genotype evaluability are negatively correlated. But what's more interesting, we never look at genotype robustness, right? We always look at phenotypic robustness. How does a particular behavior remain robust, right? So it turns out that phenotype evaluability and phenotype robustness are positively correlated. What is phenotype evaluability? It's the number of different phenotypes found in the immediate neighborhood of a given phenotype, that is the one neighborhood of all genotypes expressing the same phenotype or exhibiting the same phenotype. One immediate neighborhood, it's a network. So the neighborhood of the entire network. What is my entire zone of access? Right. So let's go back to this. So in this case, the one neighborhood of the phenotype network contains of the neutral network contains how many phenotypes? In the center contains 3 phenotypes, right? Here one neighborhood contains 0. Here one neighborhood does contain 3, but the network itself is completely disconnected. So potentially this is what is innovation. Why is this innovation? It's interesting to understand. So let's say you start here. You are a normal RNA sequence or you are a metabolic network that's just growing on some substrate and so on. You keep harboring certain mutations. So what kind of mutations are these? These are neutral mutations, right? If you stay on the network, they are all neutral mutations. If you fall off the network, they could be non-neutral mutations and so on. But what's a neutral mutation? The phenotype remains the same, so it could be some phenotype. Obviously, you're often looking at a particular phenotype. There are other phenotypes that might change, right? So because phenotype is not the whole thing, right? So you may look at ability to say grow in one carbon source. If that is your phenotype, that ability might remain, but as you keep neutrally moving there, you might find that other abilities improve or worsen, as the case may be. And this is very interestingly found even in protein structures and so on, wherein you find that there are, like, let's say you have a protein that carries out a particular function. Let's say it's an alcohol dehydrogenase. So what is important for this function? Some active site residues, right? But therefore, as long as you keep the active site constant and you change a few other things in other parts of the protein can retain its ADH function, alcohol dehydrogenase function, but at the same time mutate. It can harbor mutations in other parts of the structure, and maybe it now develops an active site that can do a succinate dehydrogenase. So it can bind a succinate substrate or something like that, right? So because this was constant, the organism survived in the first place. So it slowly harbored neutral mutations which enabled to then get close to. So this is, let's say, alcohol dehydrogenase. It's slowly moving, mutating. Alcohol dehydrogenase ability has remained unchanged all along, but suddenly it's close to picking up a succinate dehydrogenase ability. Phenotype is some function ability. Yeah. So how would you generally define a phenotype? usually function, right? And even when you say, because even if you say structure, you are always finally concerned about function being the phenotype, right? Structure is usually a proxy for function. It might change, it would change, right? So in that case, nothing would be a neutral mutation, right? So every small, but potentially, some amino acids changing may not significantly affect the structure as well, right? So you may have to consider it that way, right? Because if you then say I'm going to permit no change in structure, then no mutation can ever be neutral. Yeah. So if you're talking about a DNA sequence to protein structure, yes, obviously because of the degeneracy, there are going to be many silent mutations and so on. But even they are not essentially silent, because they can change the expression, which can finally change the function, phenotype, and so on, right? Because what sequence is there? Suppose a GGG. How does it get translated? A GGG anticodon has to bind, right? Which is carried by the GGG tRNA. If the GGC tRNA is less in concentration compared to GGG tRNA, the expression of the protein itself is going to change. Right? So neutral mutations may not always be neutral. So how do you actually define neutral becomes a little bit of a contentious point. But essentially the idea is this, that if you have a large neutral network, you can harbor more mutations without damaging the phenotype, and it also gives you the ability to get closer to newer phenotypes. Because you started off here, you can go around all along and reach some place like this, whereas here you can't go anywhere, and here you can just keep circling around here. You can still never reach one of the novel phenotypes. So because of these definitions, which are actually very reasonable, this is a very interesting paper published by Andreas Wagner in 2008, titled Robustness versus Evaluability, a Paradox Resolved. So this gave a very good resolution of the seemingly paradoxical relationship between robustness and evaluability, right? Because robustness is the ability to resist change, and evaluability is the ability to get new changes and innovations and so on. And you find that phenotype robustness and phenotype evaluability are very positively correlated, whereas genotype evaluability and genotype robustness are quite negatively correlated. So for any system, you have to answer the following questions. What is your genotype phenotype map? What are your genotypes? What are your phenotypes? So classic examples are, I take gene networks as my genotype, and phenotype is the ability to show an oscillation. Does it oscillate or not? So everything that oscillates forms one phenotype. And how are the resulting phenotypes distributed in the genotype space? Does this organization in genotype space have implications for the robustness as well as evaluability? So these are the kind of questions one asks of the system. There are many interesting papers that have been published in this space, and I will share some of them with you, you can have a look. So this is essentially a brief and quick introduction to evolutionary systems biology. It is a very important field inside of systems biology, wherein we answer a lot of questions through in silico evolution and so on, and very important for say synthetic biology even. How do you design robust genotypes or robust phenotypes and so on and so forth. So in today's video, we overviewed the concepts of a neutral network and how we perform a genotype phenotype mapping that enables us to construct and analyze the topology and the degree and the various aspects of these neutral networks. So a lot of work in this area comes from the lab where I did my postdoctoral research, which is the lab of Professor Andreas Wagner at the University of Zurich. So I suggest that you can go and look at some of those very interesting papers that are coming out of that lab. And following this, we will switch gears and move to the final topics in this course. We will look at synthetic biology, wherein we will study the different parts, modules, and how these modules have been combined in biological systems. So synthetic biology is essentially a very engineering approach to biology, where one wants to design different kinds of circuits and modules and so on. And we will also look at what are all the challenges in synthetic biology.